Hi and welcome to this reproduction video where I will go over DNA structure and protein synthesis, how proteins are made. This is only for biology triple students. Looking at DNA structure first, DNA is found inside your cells, packaged inside a nucleus. There is approximately 2.2 meters of DNA in your cells, which is mind-blowing when you think how small cells can be. This DNA is cut up into 46 strips and then twisted into a more compact structure called a chromosome in humans. If we zoom in on one chromosome and unravel it, we can see it is made up of a biological molecule called DNA, which stands for deoxyribonucleic acid. Sections of this DNA that code for a characteristic are called genes, and all your characteristics are determined by proteins. So a gene, in other words, codes for a protein. Proteins are therefore made from the recipe or instructions on the DNA. Now, DNA is a polymer made up of repeating units called nucleotides, one of which is highlighted in green. Each nucleotide is made up of three parts. There is a phosphate group, a ribose sugar, and a base that contains nitrogen as one of its atoms. There are four types of bases: adenine, thymine, cytosine, and guanine. You do not need to know their names, but you do need to know the letters A, T, C, and G. DNA structure is often likened to a twisted ladder shape, where the bases make the rungs of the ladder that you would climb up, and the sides of the ladder are made up of alternating sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. And this is referred to as the sugar-phosphate backbone of DNA. And this ladder is twisted up into a helix, or a double helix in fact, because DNA is actually two polymers of nucleotides attracted to each other by bonds, which get broken by enzymes when DNA replication occurs. This is when chromosomes duplicate prior to cell division. So here the two nucleotide polymers separate to form two template strands, and new nucleotides that are present in the nucleus line up alongside each template strand, and do so by a base pair rule, where the base adenine always pairs with the base thymine, and cytosine always base pairs with the base guanine. The reason why they pair up in this way is that A and T have complementary shapes that fit together somewhat like two jigsaw puzzle pieces, as do C and G. This occurs for both strands. I'm going to cheat a little here and just write in the complementary bases on this strand, but you need to imagine that there are sugars and phosphate groups attached to each letter. And this is what occurs in DNA replication just before cell division occurs. In protein synthesis, the two nucleotide DNA polymers separate wherever the gene for that protein to be made is located on a chromosome. Here, only one of the nucleotide polymers is used as a template to build a new nucleotide strand on. So nucleotides line up alongside their base pairs, A to T, G to C, and C to G. This last nucleotide, however, has a U on it, which stands for uracil, and this occurs instead of thymine, instead of T. And this is because in protein synthesis, the nucleotides used are slightly different. They're called RNA nucleotides, and thymine base doesn't actually exist in RNA. It is replaced by uracil. This is not something you need to remember. I am simply justifying the reason why you see a U here instead of a T. These individual nucleotides get linked together by bonds to form a polymer called messenger RNA. This then separates from the DNA template strand and leaves the nucleus and heads to a ribosome in the cell's cytoplasm. So I shall get rid of the DNA strand, and what you see now on your screen is what occurs in the cytoplasm. I'm going to extend the messenger RNA strand just for illustrative purposes here. And now this is the clever bit. Like words in an instruction manual, those words have meanings, and the sequence of letters in those words have meanings. The sequence of these base letters are effectively read, kind of like an instruction manual, by the ribosome the template is attached to. And the ribosome interprets this base sequence into an amino acid. We have 20 amino acids, each of which is coded for by a different sequence of three bases. So here we have base 1, 2, 3, and this code AGC codes for one of these 20 available amino acids. The next triplet of base 1, 2, 3, UCA, codes for a completely different amino acid depicted by a different colour. Amino acids are brought to the base triplet by a carrier molecule, which are these grey shapes here. And I will depict the amino acids as circles, and different colours represent the different amino acids. You will see similar diagrams in your textbooks. So the triplet AGC codes for this purple amino acid, and the carrier molecule collects this amino acid, and your amino acids are always awaiting collection in the cytoplasm. So when you digest a meal containing protein such as meat or dairy, your digestive system breaks the meat down into amino acids, and these amino acids are soluble and they can diffuse into the blood at the small intestine. The blood transports them to your cells for this purpose, to make new proteins. The next triplet, UCA, codes for the green amino acid, which is picked up by a carrier molecule and taken to the ribosome, where it slots into place alongside the template. A bond can now be formed between these two amino acids, and the carrier molecules are no longer needed, so they then exit the ribosome and go into the cytoplasm, ready to collect another amino acid. Although I have two amino acids here, the reality is many more get linked together, and this chain of amino acids are then folded up into a specific protein shape. A good example would be an enzyme. Enzymes have a specific shaped active site in which a substrate molecule fits perfectly into. And the shape of the active site is coded for by the sequence of amino acids it is made from, which in turn is coded for by the base sequence on DNA. And the shape of a protein is very important. In fact, it's critical for the function of that protein. But sometimes, often during DNA replication, a mutation may occur which changes the base sequence. So while A and G are okay, C may get substituted for a different base such as G. So this new triplet AGG codes for a different amino acid, and that means that the protein won't fold in the same way, the active site may morph into a different shape, and the substrates will no longer fit, and this protein will no longer be able to do its job. Many inherited disorders such as cystic fibrosis are caused by such a mutation. So in summary, a mutation is a change in the base sequence, and mutations actually occur all the time, often in non-coding parts of the DNA. So these do not code for a protein, and when mutations occur here, they often do not alter the phenotype, so they don't alter any of the characteristics of that individual. So sometimes a mutation in this non-coding part could actually switch on genes that have been silenced, such as cancer-causing genes. So even though they may not code for a protein directly, some mutations can have quite a dramatic impact on the individual, and often a negative impact. However, evolution by natural selection could only occur if there were mutations and variations in organisms. So mutations have positive and negative attributes. So that finishes this video. My advice to you would be to get some unlabeled diagrams of DNA and see how much you can actually label from memory, and go back over the information that you missed out, and perhaps convert this video into a flow diagram, like a sequence of step-by-step instructions on how the DNA could be turned into an amino acid sequence, and how that amino acid sequence could then form a protein, and what then happens if the base sequence on DNA is altered. Thank you very much for watching. Check in again soon.